speaker will be Nishanth Nair from the NIH, and he'll be talking about contribution of synthetic lethality to cancer risk and onset time across human tissues.
Good afternoon, everyone. I would like to thank the organizers for having me here for talking. This is a joint work with Koyang, Ju, and Eitan, and we are from the NIH. So tissue specificity in cancer is the rule and not the exception. For example, various genetic alterations take place in the genome. They affect, they promote cancer in some tissues, but not in other tissues. And this is an open question in cancer research. Tissue specificity of cancer is likely to be rooted in the underlying biology of tissues. So in this talk, we will be focusing on 3 different kinds of cancer tissue specificity: lifetime cancer risk, cancer onset time, and tumor suppressor genes, and why they are tissue specific. I will explain each one of them in detail. So lifetime cancer risk it denotes the chance of a person being diagnosed with cancer during his or her lifetime. We are looking at only adult cancers in this study. So in this graph, the y-axis is lifetime cancer risk. It can vary from 0 to 1, and the x-axis is different cancers. And you find almost 1,000 or a 10,000-fold change from the lowest to the highest. So for example, breast has a higher chance of having cancer, but sarcomas have rarely have cancers in the population. And we get the data from the SEER database. It's a United States data set. And you almost never hear of heart tissues having cancer. The second thing that we want to study is cancer onset time and how they vary across tissues. Again, we are looking at only adult tissue, cancers here. And cancer onset time is the age when cancer happens. So if you look at the age of the first diagnosis, the median age of diagnosis of thyroid cancer is around 51 years. And for pancreatic cancer, it's 70. So we would like to know why it is so. And the third question that we would like to study is about why certain tumor suppressor genes are tissue specific. So tumor suppressor genes, if they lose function, that can lead to cancer in some tissues. For example, the BRCA1 mutation, it's a well-known mutation. If this happens, then it can lead to cancer in breast and ovarian cancer, but not in lung cancer or other cancer types. So now, some of the prior work in the lab has been in this field, not in the lab, sorry, has been there have been a couple of papers by Tomasetti and Vogelstein in 2015 and 17 in Science, where they showed that the total number of stem cell divisions in a tissue can explain lifetime cancer risk. So in this, the x-axis is total number of stem cell divisions, the y-axis is lifetime cancer risk, and these show a pretty strong positive correlation, and these are the different cancers. So their argument was, if a tissue has a large number of stem cells, that means that it has gone through much more cell replication, and therefore there could be random errors during each replication step, and that could accumulate over time, and that could lead to cancer. As far as we know, there's no prior work trying to explain cancer onset time. Now, in this talk, we would like to ask the question, can we explain lifetime cancer risk, cancer onset time, and tumor suppressor genes through the concept of synthetic lethality? All these 3 are not completely independent concepts. They are all related to cancer development. Now, first, let's define what synthetic lethality is. It's a genetic interaction between 2 genes, gene A and B. If any one of the 2 genes is lost, the cancer continues to live happily. If both genes are lost, the cancer dies. So in this talk, when we say synthetic lethal, we also mean synthetic sick, in the sense when cancer need not completely die, but it can be just less viable if both genes are lost. There can be a dosage effect too, in the sense 2 genes do not have to have completely loose. It can have low expression, for example, rather than a complete knockout. Synthetic lethal pairs are known to be context dependent. They could be lethal in cancer, but not in normal tissue, for example. Now, this is based on prior work from our lab, from Lee et al. In Nature Communication. It was published in 2018. It's a computational pipeline called ISLE, which can mine thousands of patient tumor data and identify clinically relevant synthetic lethal pairs in a pan-cancer manner. So ISLE, it uses TCGA data, 28 cancer types, around 9,000 patients. It has 4 different steps. I will just talk about 2 of them. It looks for negative selection in the patient tumor data. If 2 genes are lost or have low expression or low copy number, they should be less likely to occur by chance because synthetic lethal pairs are bad for cancer, right? The second is if such an event occurs, then it should lead to increased survival because a synthetic lethal pair is bad for cancer, and so the patient should live longer. And they look for such naturally occurring pairs and identify a pan-cancer synthetic lethal network, which we will use for our work. Now we define what we call a CSL load or a cancer SL load for each sample. So a cancer SL load is basically the fraction of low CSL or cancer SL gene pairs. So if you look at 2 samples, 1 and 2, let's say gene X, gene A, gene Y, gene B, gene Z, gene C are all CSL pairs. And all these gene X, Y, Z are all low expressed. Even gene A, B, C have low expression. So there's a low formation. So there is 3 out of 3 is equal to 1. So an CSL load for sample 2 is 1. In sample 1, the CSL load is 0 because it's high, low, high formation. There is no low formation of these SL pairs, and so it's 0 out of 3, which is 0. So it's a number between 0 to 1. So we know that low CSLs are detrimental for cancer cells because synthetic lethal pairs are bad for cancer. So a high CSL load in a cancer sample implies that there are a large number of low CSLs, and therefore we expect an inverse relationship with cancer fitness. So we can compute CSL load for each cancer sample, and we expect a negative relationship with cancer fitness. So first we look at the TCGA data. We look at different cancer types, and we compute the CSL load for each patient. And we find a very strong negative correlation between proliferation rate, cancer proliferation rate, which we can use to the proliferation index, and the cancer SL load. So for each of these cancer types, you will find a very strong negative correlation. This is the Spearman correlation coefficient. This is a negative log p value. Most of them are on the negative side, below 0. Even if you look at individual cancer types, you find a strong negative correlation of -0.5, -0.6, and so on. We repeated this so this was using computationally identified SL network, but we also collected SLs from experiments, double knockout CRISPR or shRNA experiments publicly available. We have an experimentally identified SL network, and we also find a significant negative correlation between proliferation and SL load, CSL load. We also look at breast cancer cell lines where we have the growth rate measurements, and we again find a negative correlation. Then we also find that CSL loads are lower in the later stages of cancer than the earlier stages of cancer. So in this is a rank biserial correlation graph. It can vary from -1 to +1. It measures the effect size of a Wilcoxon test, and this is a negative log p value. For certain cancer types, you find that if it's below 0, implies that CSL loads are lower in the late stages of cancer than the earlier stages, and you find for a few cancer types that fall in that category. If you look at individual cancers too, that CSL load is higher in early stages and lower in the late stages with a significant p-value. Again, implying that CSL load is a negative marker for cancer cells. Now we take a step back and we go back to normal tissues. And we ask the question, how can we talk about normal tissues and how it affects cancer development through the concept of CSL load? So we first look at cancer versus normal, and we find that there is higher CSL load in normal tissues than cancer. So this is again a rank biserial correlation graph, and this implies, if it's below 0, implies that CSL loads are lower in cancer and higher in normal. This is a negative log p value. And you find that for separate cancer types, normal versus cancer, there's a drop of CSL load from normal to cancer. implying that some CSLs have to be lost from normal to cancer. We again find a stronger negative selection of CSLs in cancer compared to normal. So this is a level of negative selection. And if you look at normal and cancer, for example, this is random SLs, and this is the CSL network, and there's a drop on both sides, but the drop is much more significant in cancer compared to normal. This implies that CSLs are more detrimental to cancer cells and may not be so detrimental to normal cells. These are cancer-specific SL pair. So our hypothesis is as follows. Let's say you have a two loss of functions of two genes. A normal cell is not affected. A similar loss of two, if both genes have lose function, the cancer cell dies or becomes less viable. So a normal cell can have both genes low expressed or have loss of function in both genes. A cancer cell can have only one of the two. And therefore, for a normal cell from this state to become a cancer cell, it has, one of the two genes have to gain function or gain expression. And this is a malignant transformation. Now we would like to ask the question, can CSLs affect cancer development? And now we look at normal samples from the GTEx data set. These are normal tissues. They have around 6,000 samples, and we use around 6,000 samples, around 16 tissues, gene expression data, and we can compute by looking at its expression state whether the compute a CSL load for each sample in the GTEx sample data set. And we hypothesize that since that CSL load will have a relationship with cancer development and cancer risk, and we expect a negative relationship actually. So this is our important result. We compute what we call the tissue CSL load, which is taking the median of all the CSL loads of all the samples in a particular tissue. And we find a very strong negative correlation between, so first we map every cancer type to every tissue in the GTEx to the cancer. We can map that. For example, breast cancer can be mapped to the breast tissue, and we find a correlation, and we find a very strong negative correlation between lifetime cancer risk and tissue CSL load, which is the correlation is like minus 0.66 with a significant p-value. This implies that tissues which have high CSL load are less likely to develop cancer. It shows that CSL is a negative load is a negative marker for cancer development in the sense that it kind of prevents cancer from occurring because these SLs are negative in cancer, not necessarily negative in normal. So as a normal tissue becomes cancer-like, it starts becoming functional and it's detrimental to cancer. This is done for older populations. We repeated this experiment using experimentally identified CSLs, and we find a similar negative correlation. The next important result that we would want to talk about is that CSL loads can explain to some extent cancer onset age variation across tissues. So we hypothesize that for younger populations, if tissues which have high CSL load will delay the onset age of cancer because CSLs are again negative for SLs are negative for cancer, so it delays the onset age of cancer. And so that's what we find. We find a positive correlation, and this is in younger populations. So if a tissue has high CSL load, the age of onset of cancer is much delayed. And as far as we know, this is the first explanation of cancer onset age. There is no prior work on this. The third question that we would like to ask is about tissue specificity of tumor suppressors and why certain tumor suppressors like BRCA1 is specific to some tissues but not to other tissues, a mutation in such tissues. So a tumor suppressor gene when it's lost, can lead to cancer in some tissues. So here let's consider two tissues, tissue 1 and 2. Let gene S be the tumor suppressor gene, and let gene S have three CSL partners, A, B, and C, A, B, and C. Now, in the tissue 1, A, B, and C have all high expression. In tissue 2, they all have low expression. So now, if gene S is lost, or it is mutated, it forms a low-high, low-high, low-high formation here. That means there's no functional SL pair when gene S is lost. When in tissue 2, it's lost, it forms a low, 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 low formation here. That means there are 3 functional SL pairs when gene S is lost in cancer, right? And these are functional in cancer. So this implies that in this tissue where you have 3 SL pairs and SL is negative for cancer, gene S will not drive cancer in this tissue because you have 3 SL pairs that can prevent it, kind of thing. And here there is no functional SL pair, and gene S can act as a driver gene, a tumor suppressor gene in tissue 1, but not in tissue 2. So this is our hypothesis, may or may not be true, and we check it. So we get data from the cosmic data set. We can know we can collect all the tumor suppressor genes and which tissues they drive and which they don't. So we collect data, we identify synthetic lethal partners for each tumor suppressor gene. We are not able to do that for all genes. Some of the genes don't have, many of the genes do not have any SL partner, but for many of them do. Around 23 of them do. And for a majority of them, we can, if you look at the median differential expression of CSL partners, is the negative log p-value. Above 0 implies that the expression of its partners is higher in tissues that it drives, these tumor suppressors drive. And you can see BRCA1 and things, and it's all on the high positive side. Just to explain it further, if you look at 2 specific cases of BRCA1 and FAS, so if you look at BRCA1, which is a famous tumor suppressor gene, it has 4 synthetic lethal partners, and this is and we know BRCA1 drives in cancer in ovary and breast, so I'm just showing as an example ovary, and it does not drive cancer in kidney, liver, and thyroid. So if you look at the partner gene expression, it's much higher in ovary than, say, in kidney, liver, and thyroid, and this is true in general for all the partners. Similarly, for FAS gene, it drives cancer in testis, but not in brain, prostate, and uterus. And we find its partner genes have higher expression in testis compared to the other tissues. So finally, the take-home messages are as follows. Cancer S cells are selected against in cancer rather than in normal tissues, and they are lower in cancers versus in matched normal tissues. This testifies that CSL load is detrimental to cancer cells and acts as a barrier to cancer development. We see that CSL load is lower in later stages of cancer. It is also lower in more highly proliferating cancers, showing that it's a detrimental mark in cancer. We find that tissue CSL load, this is our important result, is strongly negatively correlated with lifetime cancer risk, which implies that if a particular tissue has a high CSL load, it is less likely to develop cancer. Then we also computed CSL load in younger populations, and we find that if a tissue has a high CSL load in younger populations, the age of onset of cancer is delayed. So it has a later onset age. We also find that if you look at the partner genes of tumor suppressor genes, CSL partners, and if you look at the expression state of these partners, you can explain why some tumor suppressor genes are tissue specific and others are not. So can we explain lifetime cancer risk, cancer onset time, and tumor suppressor genes, and why they are cancer tissue specific using the concept of synthetic lethality? Yes, we can. I would like to thank my advisor, Dr. Eitan Rupin, and this was a joint work with Koyang and Dr. Jusung Lee, and these are the others in the cancer data science group and our collaborators who gave some wonderful comments on the manuscript. We submitted this to Nature. It's currently under review. And we have a poster today evening. It's V056. You're all welcome to come. Thank you very much.
We have time for one quick question. Okay, in that case, it's already time. So thank you again.
Thank you.